Good afternoon. I would like to thank YIM for allowing us to present our work entitled IL-4 STAT6 signaling induces CD20 in CLL, and this axis is repressed by PI3K delta inhibitor idelalisib. My name is Palusova, and today I will present the work on behalf of my colleague Sandova, who is currently on maternity leave. This work was conducted in laboratory of Mraz. I have nothing to disclose. The precise physiological role of CD20 still remains largely unclear. We recently showed that CD20 is required for proper BCR signaling and is inducing during microenvironmental interaction by SDF1 chemokine that then binds to CXCR4 receptor. Notably, CD20 levels and also cell surface IgM levels are both induced in CLL cells from immune niche. We have shown previously that SDF1 induces just 30 to 50% upregulation of CD20, which cannot fully explain the twofold higher CD20 levels in CLL cells from immune niche. Others have shown that interleukin-4, another microenvironmental stimulus from T cells, induce IgM levels in CLL cells. Therefore, we hypothesize that IL-4 could be also inducing CD20. And indeed, we found significantly increased CD20 levels of CLL patient samples after interleukin-4 treatment for 24, 48, and 72 hours, both at protein and cell surface levels. We next compared the effect of SDF1 and IL-4 on CD20 induction and noted that each factor independently induces CD20 and their combination has a more potent effect. Next, we have hypothesized that CD20 induction by IL-4 might be mediated by transcription factor STAT6, as it is well-known IL-4 signaling effector. To validate the effect of STAT6 on CD20 levels, we electroporated CLL cells with siRNA against STAT6 or a negative control, and these results from 4 independent immunoblots were then quantified using densitometry and presented here on the graph, showing that CD20 protein levels are significantly increased after exposure to IL-4, and this induction was disrupted by STAT6 knockdown. Moreover, IL-4 effect on CD20 induction was completely abolished by pretreatment of CLL cells with STAT6 inhibitor. Lastly, we were able to demonstrate that STAT6 is directly binding to the CD20 promoter at 2 loci, and this binding is increased upon IL-4 stimulation of CLL cells. Altogether, these data show that IL-4 microenvironmental stimulus from T cells upregulates CD20 expression, leading to higher CD20 surface levels in CLL patient samples. PI3K inhibitor idelalisib has been approved in combination with CD20 immunotherapy. However, it still remains unclear if idelalisib affects CD20 levels or its function. Observation that in CLL the combination of ibrutinib with rituximab does not provide any clinical benefit in comparison to monotherapy alone, underscored the necessity to better understand the mechanism of CD20 regulations during BCR inhibitor therapy. Several studies showed that CD20 is decreased in response to the ibrutinib, which likely prevents its successful combination with rituximab. Therefore, we ask how idelalisib affects CD20 levels in CLL cells. Interestingly, in vivo data, which compared pair samples from patients before idelalisib monotherapy and after 5 or 9 weeks on therapy, showed a clear decrease in total CD20 levels, cell surface levels, and also mRNA levels. Next, we have tested if idelalisib interferes with IL-4 STAT6 dependent induction of CD20. And indeed, idelalisib treatment in vitro significantly impaired phosphorylation of STAT6 induced by IL-4, and also CD20 protein levels were decreased in samples which were pretreated with idelalisib. We have also shown that silencing of PI3K by siRNA also leads to the same phenotype, which is nicely shown in decreased STAT6 phosphorylation and also CD20 levels. This demonstrates that the specific on-target effect of idelalisib is probably the reason why the phenotype is appearing. Altogether, the data demonstrates that PI3K delta inhibition by idelalisib interferes with CD20 transcriptional activation by interleukin-4 STAT6 axis. And finally, to summarize the whole story, we have described in this study a novel CD20 regulatory axis showing that microenvironmental stimuli IL-4 is inducing activation of STAT6, which directly bind to the promoter of CD20, and this increases the CD20 expression, which leads to the induction of surface levels of CD20 receptor on CLL cells. We also demonstrated for the first time that this axis is also prominently reducing CD20 in vivo upon single-agent idelalisib therapy, which might have consequences for combinatorial therapy of PI3K inhibitors in combination with CD20 antibodies. In the end, I would like to thank to all co-authors and collaborators to our recently published story. Many thanks are also going to all members of our Mraz lab, and thank you all for your attention.